Today we're exploring the next steps in our biologically plausible vision system, diving into some of the most fascinating fundamental steps of human-like vision, how the brain detects boundaries, angles, and corners, and why this matters for building better AI. This is not a video about designing a traditional computer vision system. Instead, this project is about building a biologically plausible vision system so we can discover ideas that may improve all of AI: speech, reasoning, perception, and more. And as a side note, this information is from a recent online meeting of the Future AI Society, from which the recording is unavailable. This video covers most of the salient points. By the end of this video, you'll see neural circuits that behave in ways no conventional AI can. Learn about some of the techniques which could make AI better, and you'll understand why biology does things differently. This video includes information and ideas found nowhere else on the web that I think are truly fascinating. I'm Charles Simon, longtime AI researcher, software developer, and manager. Beyond AI, I've developed software for neurological test instruments and neural simulators. I created the Future AI Society to explore how neuroscience can inform smarter, more human-like AI. I'm using our open source brain simulator projects for simulations and demonstrations throughout this video series, and we're working on software which implements many of the learning techniques. If you want to experiment on your own, you can download the software from GitHub and try it out. If you are interested in this type of content, be sure to like, subscribe, and ring that notification bell, because the YouTube algorithm won't surface videos like this unless you ask for them. This series is documenting a project to create a vision system using biologically plausible methods. So why start with vision? Because human vision is the best studied and most experimentally understood part of the brain. Even better, many of the principles used in vision, like pattern detection, context sensitivity, and incremental learning, are likely shared across other brain regions, including speech and high-level cognition. Vision gives us a window into some general principles of biological intelligence. And by modeling these principles, we can discover ideas that will make AI faster, more efficient, and more human-like. This project isn't about creating the best vision system, it's about using vision to uncover new architectural ideas that current AI systems simply ignore. To keep this vision system grounded in reality, everything we'll build follows biologically plausible rules. That means we can use neurons, synapses, Hebbian learning, creating local changes to synaptic strength, and simulating spiking or firing rate comparisons, and a graph of nodes and edges which we've previously shown could be created in neurons and synapses. While we can't use backpropagation, matrix multiplication, global error signals, and high-resolution analog values, because these operations simply cannot be implemented by biological neurons. And timing matters enormously. For example, adults can typically read a little over 20 characters per second. This means a visual interpretation has only a few neural spikes to complete an action before the next wave of input arrives. Relative to computers, neurons are really slow, so the brain relies on shallow, fast, massively parallel circuits. We'll try to follow the same rules. Even though you could build digital logic from neurons, a neuron-based CPU would run at maybe 1 or 2 instructions per second. So the brain does not and cannot perform the kinds of deep sequential computations we use in silicon. Another constraint is evolution. The more complex a circuit needs to be, the less likely it is that evolution would have produced it. So we'll focus on simple, reusable neural motifs, the building blocks evolution could plausibly create. These motifs must handle boundaries, color values, angles, corners, and ultimately shapes. And throughout this visual processing pipeline, they must fit within tight timing limits so the system can recognize objects quickly enough to support real-time perception. We treat vision as a pipeline, even though many of the steps run in parallel. The early stages look like this. In the retina, we get raw color inputs represented by cone cell firing rates. Also, boundary detection works by comparing adjacent retinal cells. Then we'll move on to angle and orientation patches, corners and arcs, and finally, shape representation. In today's video, we'll focus on the first 4 steps from boundaries and value detection to finding corners. I'll touch on color values which follow a different pathway and lead to an interesting optical illusion. Your retina contains densely packed cone cells that fire at rates related to the intensity of red, green, or blue light. The layout, a square grid, a hexagonal grid, or whatever, doesn't matter to downstream processing. All that matters is that each pixel is represented by firing rates. Other neurons can compare these firing rates to detect boundaries and colors. But here's the twist. Boundary detection and color value detection must be independent processes. And boundary detection is far more important for understanding shapes. Let's talk about one of the most brilliant, efficient tricks biology uses, which I'll demonstrate in the Brain Simulator 2 neural simulator. Boundary detection occurs by comparing the firing rates of adjacent retinal cells. I'll show this as just one color, but you can imagine how more cells could process 3 colors simultaneously. If one pixel fires faster than its neighbor, there's a boundary between 2 colors. We can model this with a simple neural circuit with 2 pixel neurons labeled X and Y, representing adjacent retinal cells. We start with both pixels firing at a rate of 5. Assuming that each unit of rate represents 4 milliseconds, so that a rate of 1 means the neuron is firing at its maximum rate. These 2 neurons are both firing every 20 milliseconds, and there is no boundary between them. These neurons are connected to the 2 detector neurons with weights of +½ and -½ respectively. Since the inputs are firing at the same rate, there is no boundary between them, and neither of the faster detector neurons is firing. Here the excitatory synapses and the positive weights are shown in green, and inhibitory neurons are shown in black. Let's change the firing rate of X to 3 so that it will be firing every 12 milliseconds and is firing faster than Y. Now there is a boundary between X and Y, and the X faster neuron starts firing. Why? Because each boundary detector neuron receives excitatory input from one pixel and inhibitory input from its neighbor. If both inputs are firing at the same rate, the detector will receive alternating excitatory and inhibitory signals, and so will never reach its threshold and will never fire. If one signal is faster than the other, there will be occasions where 2 excitatory inputs happen without any intervening inhibition. Whenever this happens, the detector will reach its threshold and fire. The greater the difference in the firing rates between X and Y, the more often this will occur, and the faster the boundary neuron will fire, indicating a sharper boundary. Now we'll swap the firing rates of X and Y and notice that the neural circuit quickly detects that Y is faster. Here's something really important. This simple circuit can compare two firing rates without ever detecting what the firing rates are. This is radically different from how computers work. A computer must know the values of two numbers in order to compare them. But neurons can detect which is greater without encoding values at all. This is one of the ways the brain stays incredibly fast and efficient. And downstream processing like shape recognition, contour following, or corner detection can operate on boundaries only, ignoring the actual color entirely. I should add that the model I've used in this explanation simplifies the process substantially, but the principle is true to the boundary processing in your retina. We can easily imagine that a similar circuit could detect changes in a sound level necessary to recognize speech. Interestingly enough, an identical circuit could be used as a visual motion detector. Instead of detecting the difference between the firing rates of one pixel and its neighbor, it could detect the difference between the firing rate of a pixel now and the rate of the same pixel a few milliseconds ago. I won't go into how this can be done right now, but it is an example of how a single neural motif can be used for different applications in the brain. Now let's look at the other half of the problem, detecting the actual value of brightness or color. A different kind of neural circuit is required, one where each neuron corresponds to a specific value or range. This time it takes advantage of the neuron's inherent leakage rate. If two input spikes are received before the neuron's internal potential leaks away, the neuron fires. This happens if the input signal rate is at or above a specific detection rate, which is governed by a combination of the incoming synapse weight and the neuron's leakage rate. In this implementation, I've added inhibitory synapses, so the fastest detected rate will inhibit the firing of lower frequency detectors. You can see this in action if I change the input firing rate from 4 to 2. This is obviously more expensive biologically than boundary detection because you need at least 2 neurons per detectable value or value band, and the precision comes with a cost because the more different values you want to detect, the more neurons you need. For efficiency, the brain may not care about extremely fine resolution. In practice, this means the brain's visual system maintains good boundary detection, but lower quality color precision, a trade-off that favors shape over exact color. And this brings us to the gray level illusion. You've probably seen this visual illusion. Several rectangles appear to have a gradient from left to right, even though no gradient exists. The illusion demonstrates the fundamental idea that your visual system detects boundaries far better than it detects absolute brightness. When the rectangles are adjacent, you see a strong boundary. At the same time, it detects little or no difference in the gray values of the two rectangles, so the brain infers a gradient. When the rectangles are separated, the illusion weakens or disappears altogether. This is because your brain detects boundaries which are fast and accurate, but brightness levels are coarse and ambiguous. So your brain effectively says, since boundaries exist but the gray values are unclear or the same, the best logical explanation is that brightness must gradually change within the rectangle. This mechanism also explains why a wall lit from one edge still appears to be one consistent color, even though the measured brightness differs dramatically across the surface. The brain largely ignores absolute brightness because boundary-based interpretation is what actually matters for understanding shapes. Now, the simple circuit I showed for boundary detection is clearly not learned, but is hard-coded by your DNA. The value detector is a bit more flexible in that we know that an infant's color perception improves dramatically over the first year. This implies that the value detector circuit starts out with a minimal hard-coded structure, but over time, additional values can be added to the color detection ladders. In the first case, no form of Hebbian learning or AI mechanism seems applicable. And for my downstream software development, I've used a completely different boundary detection algorithm, which arrives at the same destination. For color values, we could speculate that your brain is hard-coded by DNA to detect perhaps 8 levels each of red, green, and blue, but has the capacity to learn to differentiate these values by a Hebbian process I'll describe later. I haven't pursued this as yet because I've focused on boundaries, but it is interesting to speculate that perhaps your brain can represent fewer than 512 unique colors. But coupled with the ability to detect boundaries, it gives the same unlimited color perception that you get through that gray level illusion. Now that we've explored how boundary and value detection fit into the visual pipeline, we can move downstream to the next major step. Once we have reliable boundaries, shape detection and object detection can operate almost entirely on these boundaries, and color information still matters, but it can be added later through a different pathway. The next step occurs inside the visual cortex, where certain cells respond to specific angles of very short boundary segments in the visual field. These angle-selective cells aren't theoretical. They've been found and observed experimentally. Their firing rate curves, as shown in these graphs, indicate how strongly a particular neuron responds depending on the orientation of a line passing through its receptive field. Some cells fire at up to 60 Hertz at their preferred angle. And when you think about how long it takes a neuron to accumulate enough evidence to choose which angle is present, you begin to see how this imposes strict limits on the overall speed of the visual system. In the Brain Simulator 3, I've built a demonstration of how angle detection can emerge using simple local learning. Imagine a visual field where each pixel represents either the presence of a boundary or the absence of one, a completely binary input. In this case, my visual field is 15 by 15 pixels. As we progress further into the recognition process, we'll need to expand this, but as for now, we're just looking for lines, and in the next section, corners, and this is sufficient. At each pixel location, there are 8 small patch cells, and each patch cell will correspond to a different angle. This means that at every point in the visual field, there are 8 possible local interpretations of how a boundary segment might be oriented. In this demonstration, each patch cell considers inputs from the pixel positions it represents and an area surrounding it, in this case a 5 by 5 square of pixels. In this demonstration, I'm representing pixels and patch cells as nodes in the Universal Knowledge Store, or UKS. Patches are connected to pixels with a has-boundary relationship type with weights which adjust over time. In a moment, you'll see that the 8 patches operate in a winner-take-all fashion. Only the most likely angle, the one receiving the strongest or best matching input, wins. For example, the underlying boundary is horizontal, and one patch will have incoming weights which make it fire the strongest. In the event of a tie, the tie is broken randomly using the first patch in the relationship list. If I right-click on a pixel position, we can see these 8 patch nodes at a specific location and the color-coded input weights for the surrounding boundary pixels. Initially, all patches have identical synaptic weights. Each patch is most strongly influenced by the pixel at its center, and the weights fall off radially in a roughly circular pattern. In this demonstration, the patch is a 5 by 5 square, but in biology, it might be larger and circular. As training progresses, simple local weight adjustments begin to differentiate the angles, and you'll see how the weights approximate them. After 10,000 input lines, you can see that the weights of the 8 patches of a single pixel are beginning to represent different angles. In the display, I've added little black lines to indicate the preferred angle based on the weights of the relationships. And you can see that the sets of 8 patches generally organize themselves so that they can recognize 8 different line angles. A key idea here is that although nature contains very few perfectly straight line boundaries, any curved or jagged boundary, when viewed close enough, is composed of small segments that are straight across a few pixels. Because of this, the entire patch training process I'm using relies exclusively on straight line examples. After 10, 20, or 30,000 training examples, each set of 8 patches gradually self-organize so it becomes sensitive to a specific angle that appears most often in its training situation. This mirrors what we see in the biological visual cortex. Which node represents which angle is random because of the random ordering of the input. I originally assumed inhibitory synapses would be important. It seems intuitive that if the correct line is present but extra pixels also fire, those extraneous inputs should negatively affect the recognition. But I found that over time the competitive nature of the 8 patches at each location eliminates the need for inhibitory connections entirely. Any incorrect interpretation is naturally outcompeted by the patch whose pattern better matches the input. Another important observation is that we can remove synapses whose weight drifts close to 0. If a connection then contributes nothing, eliminating it entirely matches biological efficiency but also dramatically speeds computation. This selective pruning moves the architecture closer to a sparse, efficient, biologically inspired network, unlike typical artificial neural networks which process dense arrays with weights even when many of those weights are 0. It's also interesting to consider how this learning process compares with a biological timeline. Research suggests that newborns don't begin life with pre-wired angle detectors. Instead, angle sensitivity develops gradually over months or years. With 8 angle detectors, the system I've built probably resembles the visual capabilities of a 1-year-old. A child younger than that cannot possibly read, in part because stable angle perception hasn't yet fully developed. From this, we can conclude that angle detection is learned, not pre-wired, unlike boundary detection, which appears far more innate and hard-coded. This distinction is critical. Some parts of the visual system are genetically programmed, while others must be shaped by sensory experience. Another conclusion emerges from the limited number of angle detectors. As an adult, if you can only detect a dozen or so discrete angles, then smooth curves and tiny variations in orientation aren't actually detected directly. They're inferred. The brain applies the same logic it used in the Grayscale Illusion. When neighboring pixels detect slightly different angles, the simplest explanation is that a curve is present. The brain fills in that interpretation even though it has never directly detected a curve at the pixel level. Curvature becomes an emergent property of the angle detection system rather than a primitive feature. All of this sets the stage for even more sophisticated processing. Once you have local angle detectors, simple, sparse, competitive, efficiently learned, you can begin to build corners, junctions, and eventually coherent shapes. And just like angle detectors, these higher level features will emerge naturally from the structure of the system rather than being programmed directly. Now that we've explored how the system learns to recognize small line segments and the angles at which they appear, the next step along the path towards recognizing objects in a visual field is the ability to recognize corners. Corners emerge naturally once we begin to connect the angle detector patches into a larger rotational structure. And this is where the universal knowledge store, our graph of nodes and relationships, becomes central to the process. To begin, we introduce a new relationship type in the UKS, collinear width. When the system is shown a straight line, only one patch at each pixel location will be the winner. Because those winning patches all represent the same local angle, we can treat each pair of adjacent winning patches as collinear, and we can add or strengthen that relationship between them. This gives the system a way to represent continuity of a line in a relational form. With this mechanism in place, corner detection becomes surprisingly simple. A corner occurs whenever two adjacent winning patch nodes do not share a collinear width relationship. In practice, this means that the moment two neighboring pixels both have active angle detectors, but these detectors represent different angles that cannot be aligned into a single straight segment, the system identifies a corner. Alternatively, we could say that the existence of a collinear width relationship inhibits the corner detection. As we present the system with corners, the detected corners are shown with small green squares. Even at this early stage of software development, this method detects corners with striking accuracy, as you can see in this demonstration. Every so often a corner is missed or an extraneous point is marked as a corner, but on the whole, this works surprisingly well, and there was no training about corners whatsoever. This idea is still new and not yet fully fleshed out, but the results are, so far, extremely promising. Just like patch training, these collinear width relationships learn their strengths using a Hebbian rule. The weights associated with collinearity form in exactly the same way as the patch synapse weights that established angle detection. As the boundary detection and angle detection layers become more refined, the system's ability to detect and connect corners continues to improve. Before moving on, it's worth reviewing several important conclusions. One is that boundary detection appears to be a specialized, biologically pre-programmed capability. In contrast, angle detection and corner detection seem to be learned capabilities, general-purpose mechanisms that arise from repeated exposure to patterns in the visual field. There are deeper implications here about how the brain represents information. Even though we can determine that two angles are collinear or not, we still don't know what the actual angle values are in absolute terms. This highlights an important biological truth. Almost everything in the brain is relative, not absolute. Visual up and down are not fixed reference directions tied to the world. They may be defined relative to other directions or by the inner ear, but down typically just means in the same direction as my feet. Likewise, in the visual cortex, the image could be upside down or sideways, and the angle detection and corner recognition and even object recognition will work in exactly the same way. The addition of the collinear with relationship is also interesting for another reason. This kind of relationship is essentially impossible in traditional artificial neural networks. Standard ANNs rely on computation that moves strictly from one layer to the next. They do not allow dynamic learned relationships among units in the same layer. Yet biological networks do make such lateral connections, and they appear to be essential for capturing structural relationships like collinearity. It may turn out that these lateral relationships are necessary for advancing artificial networks beyond their current architectural limits. Finally, this leads into a feature that we will need going forward to recognize arcs. And we're adding the relationship type nearly collinear with. Instead of only marking patches that are collinear when their angles are both winners, we can also form relationships between a patch and the next best angle candidates in the same cluster of 8. Each angle detector responds to a range of angles, but responds most strongly to its center angle. Angles near that center will also respond, just not as strongly. So we can connect the strongest to the second and third strongest firing nodes with a nearly collinear with relationship. After learning, this imposes an ordering on the set of 8 angle detectors. While not yet complete, it initially appears very promising. And it's essential for the next phase of the project because arcs and curves require more than simply knowing when angles differ. To detect an arc, we need to know when angles are not collinear, but also when they are close or almost collinear, just slightly off from one another. That subtlety is what will allow the system to distinguish between a sharp corner and a gently curving boundary. This ability to represent both collinearity and near collinearity will serve as the foundation for recognizing curves, arcs, and eventually more complex shapes, all using biologically plausible neuron-based mechanisms. But that's not all. The ordering mechanism is not only useful in vision, it could become a general ability throughout the brain for organizing any set of competing alternatives. Furthermore, it implies a system for representing the order of any group of things in the brain, values, distances, even time. To me, this is a really exciting direction for development. So what does this all mean for artificial intelligence? Today's AI models rely on backpropagation, high precision weights, massive matrix multiplications, and deep stacks of layers. Biology uses none of these. Instead, it uses simple circuits that detect boundaries without knowing values, operate with ultra-low precision, run in parallel, and work within strict timing limits. If we adopt biological strategies, especially these boundary-first mechanisms, we may discover AI systems that are dramatically more efficient and more capable of real-time adaptive behavior. That is the purpose of this entire vision project. This is just the beginning. In future videos, we'll explore how these fundamentals described in this video can be used for more general shape and object recognition. All this ties into the universal knowledge store, so the system will be able to understand what it sees. I hope you'll join us on this journey toward building AI that doesn't just process information, but actually understands it. If you'd like to follow along, be sure to like, subscribe, and hit that notification bell because the YouTube algorithm won't surface videos like this unless you ask for them. And then try out the software or join the community for free to participate in our online video conversations and our Discord server. Check out the links below. And as always, thanks for watching.